She's also the director of UCT's newly accredited Human Evolution Research Institute. Her research uses cutting-edge quantitative techniques to examine skeletal variation in fossil hominins, humans, primates, and a few other mammals. She's also involved in a number of projects merging these data from other information from the genotype and the phenotype. This work is aimed at understanding how evolutionary processes such as adaptation, hybridization, and even random chance can change organisms and their evolutionary trajectories through time, and ultimately the evolutionary fates. Rebecca is also an NRF B2 rated researcher, and she's the recipient of a Distinguished Teacher Award at this university. Please welcome her. Very impressive. Thank you. Okay. Excuse me while I get clipped in here. I don't think you really need me because I'm quite loud, but they're recording it. They're recording it, so let me use this thing. So which one do I want? Oh, shall I leave it like that? Yeah? Okay. Or do you want to see me? I'll be right here in front, I promise. Okay, so this is really fun for me to talk about because I have quite a wide research program, and I got to, I'm really very loud, aren't I? I got to speak about genetic drift and my work on hominilady and some other things. Ooh, yeah, turn that down, because I'm very loud. Last week to ArcSoc, and today I really get to focus in on one of my favorite topics, which is sex. And so I'm really glad you're all here to share it with me, so to speak. Okay, so what I'm really going to talk about today is, for the most part, I'll briefly mention something else, but I'm going to primarily focus in on our lineage. And when I say our lineage, what I mean is the genus Homo. So our lineage since we diversified from other hominins, things like Australopiths, right? Things like Australopithecus sativa and Australopithecus africanus and all sorts of ones you've learned about in the past. Now, our lineage is very diverse. There are many species within our lineage. This is highly contentious. People can debate for their entire careers about what species are within it. But just sort of in brief, we have everything from Homo habilis to Homo erectus to Homo heidelbergensis, Neanderthals, modern people, and then some of these little enigmatic ones with small brains, Homo naledi being one of them, and Homo floresiensis another. So there's quite a lot of diversity within our lineage. Now, our lineage was first discovered in the 1800s with discoveries of Neanderthals, but our most ancient members of our lineage, so tracking, if you will, the beginning of the emergence of Homo, was a discovery made by people you might be familiar with, Mary and Louis Leakey, in 1960. Technically, it was their son who discovered it, and it was this mandible here, which actually stands as the type specimen for Homo. So it's the specimen against which we judge all else, if you can believe it. It's just a mandible. Now, this was a specimen that formed the holotype, the type specimen for the species Homo habilis being named, and frankly, Homo generally being defined as being handy person, so habilis being handy, in other words, being a tool maker. More recently, we've actually pushed back the time. So the first discovery of the earliest Homo, of Homo habilis, was about 1.75 million years old. And recently, we've pushed back the time to the emergence of Homo with fossil discoveries in Ethiopia. And the oldest one, which was just discovered about a year ago, is now 2.8 million years old. So our lineage begins at approximately 2.8 million years, perhaps older, but we don't have fossil evidence for that. Following that, at about 1.8 million years or so, after Homo evolved for a little while, we left Africa. So everything at the beginning of the emergence of our lineage was within Africa, but at about just shy of 2 million years, Homo started to leave Africa and disperse across the Old World. So by the Old World, I mean Europe, Asia, eventually by about 50,000 years getting into Australia, and only in the tens of thousands of years ago getting to the New World, the Americas. So it's a fairly rapid dispersal that we see during that time period on geological scale. Now, as I was saying, Homo we've generally thought of as a lineage that's defined by three things. tool use, big brains, and a body from the neck down that's essentially like us. So not 100%, but essentially walking bipedally in a striding manner in a way that's similar to us. Unfortunately, to break it to you, this has all fallen apart in the last 5 years or so with a whole bunch of new discoveries. We just recently, again, in the last year, had discoveries of early stone tools, which have pushed the beginning of early stone tools back by 500,000 years, older than the oldest Homo that we have, so in excess of 3 million years ago. So we know that stone tool use is not unique to Homo anymore. Kind of gets rid of that name, Homo habilis. We also have very small-brained Homo, as I already mentioned, so Homo floresiensis, Homo naledi, some Homo that's in Dmanisi are very small-brained. So we know that even these tool makers were making them without the cognitive abilities of a large brain. And finds like Homo naledi are showing us that from the neck down, they really don't always look just like us. So from the waist down, something like Homo naledi is quite similar to us, but it has very ape-like arms and particularly ape-like shoulders. So what this is telling us is that our sort of simplistic notion of a large-brain tool-making biped on a savanna environment is just not right. And what we actually have is a very, very diverse lineage, both behaviorally, morphologically, from the head to the toe. So if you look at Homo in the context of time, this little green box is sort of outlining the time period of 2.8 million to the present that shows a lot of Homo. There are a few other things in here as well. There's a bit of Paranthropus. There's a bit of Australopithecus. But one of the things I just want you to notice as you're looking at this is that a number of these diverse species of Homo live at the same time. And for this first part, this sort of 2.8 to, well, quite a while, a lot of these species are living in Africa, so in the same place at the same time. And here we are living in different places, not just in Africa, at the same time. But there's a considerable amount of overlap between these taxa, which of course kind of brings me to the topic of today, right? If you have overlapping taxa, what does that mean? Are they distinct? Are they separate in their environments? Are they interacting? And if they're interacting, are they interacting by exchanging genes? This kind of might seem strange to a lot of people because we were taught, or many people were taught in school, that if you have a species like Homo erectus or Homo habilis or Homo heidelbergensis, that it's a discrete unit and that it's biologically distinct from other species and by definition cannot, I don't know what that is, by definition cannot interbreed with other species. Excuse me while I sort this out. For some reason it wants to open the internet for me. You can tell I lecture every day. I'm used to these sorts of things getting in the way. So we're used to this idea that these are discrete entities and that there is no gene flow, right? There is no hybridization. There can't be by definition. But the reality is, over the last decade or so, our understanding of the importance of hybridization among all sorts of organisms has grown tremendously. People used to think it was prevalent only in plants. We now know it's quite prevalent in animals, in mammals, and in primates. So if you look across primates, gene flow between distinct species, between distinct lineages, I like the term lineage because species is problematic in different ways. But between these very distinct groups is quite common. It occurs about 10% of the time in about 10% of primate species. And we've also learned from the research coming out of the gene flow, the hybridization literature, that there are a lot of different consequences. So again, we used to think about it where if you had two species and they started exchanging genes, they'd basically become just like each other, and that was the end of the story. But we now know that that's just one possible outcome. Yes, you can get this merger of these lineages, but you can also have genes essentially be exchanged and cause one lineage to go extinct or the other. You can have the production of hybrids cause reproductive isolation between lineages. You can have hybrid zones that form and remain stable in perpetuity sometimes. I mean, it goes on forever and ever. Baboons are a good example of that. But the ones I want you to kind of lock your eyes onto down here are these last two. We also know that gene flow, the hybridization and the gene flow that occurs from it, can cause really remarkable results. It can cause novelty. It can cause new phenotypes. And the reason for that is because when you have lineages coming together, you have a recombination of different things that are distinct. And you will get, sometimes, organisms that are quite distinct from their parents in different ways. They can be mosaic, they can be intermediate, they can be transgressive. They can have aspects of the phenotype that are outside of the range of their parents. And what that does is it allows evolution, selection in particular, to act on that variation in new ways. So, I'm going to talk about what we know about this in the human lineage, what we know about the role of gene flow. And when I say hybridization versus gene flow, just to be clear, hybridization is the process of coming together. Gene flow is the stuff that happens when you come together. So it's the actual flow of gene or gene exchange. Now, we have information on this in the human fossil record from really two sources, from genetic information and from information that we've learned, and this is mostly my work, from applying our understanding from living organisms to looking at the fossil record and interpreting the fossil record. So we have it from genetic evidence and from morphology, from what we look like, what hybrids look like. And I want to really focus in particularly on the recent time period, and particularly this question of modern human origins and what gene flow means in the context of our understanding of modern human origins. So just to give you a little bit of background on this, up until very, very recently, and in many people's minds, this still holds the day, the sort of dominant view of how modern humans emerged in this world was essentially a replacement model. So in other words, when Homo left Africa 1.8 million years ago and populated the rest of the world, and we had Homo erectus in particular all over the place, and then Homo neanderthalensis, or Neanderthals in this part of the world, and others, they kind of hung out there. Africans stayed in Africa, and then at some time around 200,000 years ago to 100,000 years ago, it depends, depends on who you ask, these humans in Africa evolved into modern humans, moved out of Africa, and replaced everything else. That's the replacement scenario of modern human origins, and it has been the dominant model for the last 20 years. There have been other models, but they have not been the dominant model. This is the one that's really been accepted. At the core of this model is are the Neanderthals, essentially, because the Neanderthals are one population that we have known for, or one species that we have known for hundreds of years. As I said, our earliest Neanderthal knowledge started in the early 1800s. We know a lot about what they look like. We know a lot about their behavior. We know that they look different from the fossil material that's in an African context. And we know that if this scenario is true, and people were leaving Africa and replacing fossil populations, fossil species in the rest of the world, that they would be going through that and right through the Neanderthal range, and they would have replaced Neanderthals. So at the core of this has always been what happened, the question, what happened to the Neanderthals? Were they replaced or not? So people started to ask this question, and of course, if they had hybridized, they weren't replaced, right, by definition. So people started to ask this question based on the fossil record. Yes, they look very different, so everyone was assuming that they were different and a distinct species and replaced, but also based on the molecular record. So the very first work that started to come out on this, on ancient DNA analysis of Neanderthal specimens, was on mitochondrial DNA. So not your genomic DNA, not the stuff that's makes up your chromosomes, but the things that are the little mitochondrial circles of DNA, the little powerhouses that power your cell. There are all sorts of reasons for this. It's largely because it's much more abundant in the fossil record, and so given where the technology was at the time, it was easier to recover it. So a lot of the work was done on mitochondrial DNA, and there were many specimens from which mitochondrial DNA was retrieved that were between the ages of about 30,000 and 100,000 years old. So the question became, if we compare that to people who are alive in the world today, can you see any evidence that that DNA has existed in, or exists in modern populations? And this is basically what came out of all this research. First of all, people realized, well, it's a pretty good sample, but that the Neanderthals are quite variable and comparably variable to modern people, that the Neanderthals are much more like each other than they are to modern people. Not actually all that big of a surprise, right, because they lived a long time ago. and that there's no clear evidence, and this is the real key one, for uniquely Neanderthal mitochondrial DNA in modern humans. This was a real nail in the coffin for any other ideas outside of a replacement model. And really, once this genetic evidence started to come out, people said, that's it, it's definitely replacement. Everyone emerged from Africa, that's the end of the story. Until this. This paper came out in 2010, and this is the first analysis, not the only analysis, but the first analysis that came out of a lab that's been working very hard to reconstruct entire genomes, entire ancient genomes of species with a particular focus on Neanderthals. And this first paper was based on fragments of 3 individuals from Croatia, these 3 down here, that were about 38,000 years old. Since that time, there have been 2 other Neanderthals that have been sequenced, one in Russia and one in Spain. And they have essentially said that the replacement model isn't true, that there is evidence for Neanderthal DNA in modern people today, and therefore we are hybrids, if you will, or the result of admixture between Neanderthals and modern people. I'll come back to that. In addition to those Neanderthals that were analyzed, there have been a few other individuals that have also had ancient DNA retrieved from them that aren't Neanderthals. 3 of them are from a group called Denisovans. We have no idea who Denisovans are. Denisovans live in this cave in Russia. They're not very well dated, something like 80,000 years ago. And the only evidence we have of them are from this little finger bit, a tooth, and a little bit of another small bone. So we don't know anything about them. But the ancient DNA work has shown us that they're actually quite distinct from Neanderthals, and they're quite distinct from modern people. So we know there's more diversity out there at that time, and we have the genetic evidence for it. And again, I'll come back to what that means later on. We also have evidence from an individual from Romania, that's around 40,000 years, genetic evidence, genomic evidence, that has always been called a modern human. So a anatomically modern human, not a Neanderthal, not a Denisovan, a modern human, except by one person who always questioned its affinities. But it's a clearly modern human-looking individual. So this one has also played into the picture as well. And again, I'll come back to this one as well. So essentially, what this information is now telling us is this. We have a separation genetically between a lineage that leads to modern people, this blue one on this side, and a lineage that leads to these other 2 groups, the Neanderthals and the Denisovans, on this side, and it diverged at around 600,000 years ago. Following that, we have a divergence between the Neanderthals and the Denisovans at around 400,000 years ago or so. And then we have a bunch of cool stuff going on. We have genes flowing, gene exchange from modern humans into Neanderthals at around 100,000 years ago. We have genes going from Neanderthals into modern humans at around 50,000 years. We have genes going from Denisovans into certain groups of modern humans, also around 50,000 years ago. We have genes going from Neanderthals to Denisovans. We have genes going from an ancient population that we don't even know what it is into Denisovans. So there's actually quite a lot of gene exchange. But think about this. How many individuals is this based on? Less than 10, right? So this is the tip of the iceberg for what might be going on. So this is fantastic. It's really exciting, wonderful. We're learning that gene exchange has played an important role in our origins. But it's limited in many ways. One of the ways in which it's limited is what I was just saying, sample size. It's very difficult to get this ancient DNA out of material, and it's technologically demanding, and a lot of the material, by the time you get to the time of Neanderthals, is simply too degraded to get information out of. So it's limited in terms of the time depth, the amount of individuals, and the time depth that we can look at. So we've got good information on Neanderthals. We'll probably have more. We're not going to have hundreds. We're not going to have thousands. We're definitely not going to have thousands because we don't have thousands of Neanderthals to get it out of. we can't get information further back in time. There are limits to our ancient DNA abilities. So the oldest ancient DNA we have right now is from a 700,000-year-old horse. It's very unlikely that we're going to be able to answer any questions using DNA about early Homo, Australopithecines, most of the human fossil record, frankly. So while the ancient DNA results have been incredibly exciting, and again, I'll talk about what they're saying a little bit more later, there's another part to this picture, and that is, beyond that, how can we also supplement it with the morphology? How can we figure out what these hybrids would have looked like? Excuse me. So this is where a lot of my work comes in. Over the last decade, I've been doing all sorts of studies of all sorts of hybrids, everything from baboons and marmosets and macaques and gorillas to things as distant as wildebeest hybrids, and more recently, a lot of laboratory mouse experimentation with hybridizing mice. And my goal has been to figure out what these hybrids look like. Most of the work I've been doing is on the skeleton, but I've also been doing quite a lot of work with the genotype and phenotype as well. I'm not going to go into those, the external phenotype as well. I'm not going to go into those in great detail tonight. So just to kind of sum up in a few slides, I love this, sum up your research in a few slides, what this information is telling us. It's basically telling us two things. It's telling us that hybrids are very variable. Particularly if you're going to be sampling a hybrid zone. These are baboons that come out of a hybrid zone in Malawi. There's a huge range of variation in hybrids. So hybridization, by almost definition, unlocks a lot of new variation. This is some data from mice. One of the things that happens when you have hybrids, and these are first-generation hybrids, so the two parents producing the first generation of hybrids, is you get a big bounce in body size, but you also get changes in terms of facial morphology. Hybrids tend to have larger faces across a number of these organisms that we're looking at. You can see in this picture, this is just showing the two parent groups for a lot of different crosses we've done in green and orange and blue, and the pink in each one of those is the hybrids. So they're kind of intermediate to, but distinct from, the parent groups in every case. So we can turn back to the fossil record and say we're looking for populations of highly variable, large, intermediate to, but distinct from individuals. This is some data from baboons. So again, the blue here are the F1 hybrids, and essentially what this is showing, shape is summarized on this y-axis and size is summarized on that, is that the hybrids are shifted over. So for a given shape, they're quite large, they're quite extreme, and we get these individuals that really stand out. So you can really see individual hybrids in the fossil record as well. Those ones that I just showed you are all first-generation hybrids. When we move beyond it, and this is where the variation really starts to come in. So these are parents, and these are their hybrids, and it's the same here. That's the same parents, and that's the same hybrids in the first generation in blue. This is the second generation. This is a back-cross strain. So you get all this huge range of variation if you're sampling a hybrid zone or a hybrid population relative to the parents. So again, this is the sort of stuff that we can start to apply to the fossil record. There are those hybrids there. There are the parents. So the one thing is they're quite variable, and we are getting a real grip on what the shape variation also looks like in these populations. The other thing that we've learned from these studies is that when you have two genomes coming together that are similar but distinct, because they're different species, right? Different lineages, little glitches happen in development. Because things don't quite go as they should when those genomes are coming together. It's not massive. It doesn't cause the individuals to be infertile or to have any problems, but they're little signatures, if you will, of these hybridization events. And this is one of the most exciting things that this work has shown. So things like supernumerary teeth, extra teeth in the tooth row. There's one in baboons, there's another one in gorillas, because dental development has continued on longer than it should because of these glitches. Additional sutures, that one should not be there in the face of that baboon. That one should not, those should not be there in the face of that ruminant, that wildebeest. So little small indicators of hybridization at extremely high frequencies. And when you talk about something like these supernumerary teeth, first generation hybrids, 50% of them will have these. And sometimes you get these really, really strange anomalies. So not ones that you see really at all in modern populations. So this is showing developmental problems or developmental glitches, if you will, in wildebeest. You can see this sort of strange curvature of the horns. These are double canines. This is a captive population that I've worked with. This is a wild one. Those are the only two that have ever been described ever. So you can see the two canines. This is a os parietal dehiscence. It's divided parietal bone, it's cranial bone, and it's bilateral. It's on both sides. There's only ever been one of those in the literature before. So we get these very strange things as well, which is great because if you see these, they're big red flags. So what does this tell us about the human fossil record? Well, and this has been my goal, right? How to apply this morphology to the human fossil record. I told you I'd say a little bit outside of Homo, and I am going to say a little bit outside of Homo because I'm involved in other research on Australopiths as well, looking at some of these questions. One of the places, I think, where there is good evidence that gene flow has affected our evolution is at the time of Lucy. So Australopithecus afarensis, living in Africa just in excess of 3 million years ago. Interestingly, right around the time when we now have stone tools, and just following this when Homo emerges in our lineage. Lucy is just one representative of her species. We have lots of them. It's one of the most well-known and one of the most well-known and abundant assemblages of a fossil hominin that we have. And in addition to being one of the most variable hominins that we have, people have talked about this for decades, that there's a huge amount of variation, that it should be split into multiple species. We don't, it's way too much. What is it? Is it dimorphism? So it's hugely variable, so it fits the model in that respect. There are also more than one individual that have supernumerary teeth, some of which are bilateral, which is extraordinarily rare. This one is not published, to my great irritation with the person who found it. And there's one other one that also hasn't been published. So I think there's good evidence at this point in human evolution. But probably the best evidence we have actually coincides with exactly what the genetic data are telling us. So at the time when humans were coming out of Africa, contacting people in the Middle East, and when we had also Neanderthals around the same time period, not so far away from that, we have considerable evidence for these kinds of traits, high levels of variation, asymmetry in some of these skulls, different morphs within them. We have dental anomalies, rotated teeth in particular in these different specimens. Certain specimens that have really tiny teeth, other specimens that have really large teeth, which are the same sort of signatures that you see when you get extra teeth, right? Continuing of development. And we have this individual. You've seen a picture of this individual before. This is an individual that we identified on the basis of morphology as being a hybrid. This is not the skull and the mandible aren't from the same one. We identified one of the pieces as being a hybrid, and the genetic work was actually done on the other piece. And we identified it on the basis of its tooth morphology, and they identified it as a hybrid on the basis of its genome. So it's actually a fourth generation hybrid. So this is one of the nicest pieces of confirmation that the genetics and the morphology are actually saying the same thing. So it's quite prevalent, is what I'm trying to tell you. There are other fossils that I just wanted to throw in here as a bit of a teaser as well that shows some kind of strange things. One of them is Homo floresiensis, the hobbit that lived between 60 and 120,000 years ago in Indonesia, caused quite a splash in 2004. You might remember when it was all over the papers. And it's very strange. It has these very strange bilateral rotated teeth, developmentally really unusual. Do we know it could be developmentally unusual because it's this very small creature, or that maybe there's some gene flow happening here? Certainly at that time period, there's the possibility of gene flow because we had people in that area at the same time. And I had to throw this in because I've also been part of the Homo naledi team, this is also a really strange species, this. It has this mosaic of traits, this Homo-like bottom and this more ape-like shoulder. We don't know how old Homo naledi is right now. It doesn't have a date put on it. It could be at the beginning of Homo. It could be fairly recent. But at any of those time periods, these sorts of questions are certainly worth investigating in greater detail. Okay, so what does this tell us? First, big picture, what this is telling us. Well, the take-home message here, essentially, is that the replacement model of modern human origins, a full replacement model of modern human origins, is just not tenable anymore. That gene exchange has refuted that. It doesn't mean that hominins that were in the other parts of the world couldn't have little pockets of them gone extinct or been replaced. But as a full model for modern human origins, it's no longer tenable. The second major thing this tells us is it really messes around with our ideas of species and our desire that humans have to compartmentalize everything into taxa. You know, looking at hybrids and gene flow has not been a popular thing in paleoanthropology because we tend to want to do this, right? You've got a beginning species, you've got an end species that lives today, and so every time you have a branching event, something has to go extinct because only one of those lineages could have led to us. Right? So you have a whole lot of extinction with one final end product. That's been our model for what human evolution essentially looks like. That's not what this is telling us. This is telling us that our model is possibly quite deep in time one of gene exchange. And when you have gene exchange, it's very hard to parcel taxa into these discrete units. Very difficult to do so. So it challenges kind of how we've organized the world. And the third thing that it does is it challenges the adaptationist framework. So this is also, if you will, I hate to use the word paradigm, but it really is. The way that most differences when we look at the fossil record have been evaluated is if you see difference, it means that something is adaptively different. So in other words, selection is acting to cause that difference. Now we know that both gene flow and genetic drift, which I haven't talked about today, but it's another one of my favorite evolutionary forces, act to diversify and have caused a lot of this variation that we're seeing in the fossil record. And they can even be forces that are producing novelty. So when we see really new, interesting things that themselves become adaptively successful. Okay, so coming back and to what this gene exchange has been telling us, essentially, in detail. I'm going to come back to the genetics and wrap up here with the genetics. So just, first of all, the amount of gene exchange that we have is not trivial. It's something like 1 to 4% of non-Africans have Neanderthal genomes. You notice I say non-Africans, right? You have people moving out of Africa and interbreeding with the Neanderthals outside of Africa. So you do have people left behind in Africa who didn't interbreed with the Neanderthals. And this is actually how this was discovered, by comparing the Neanderthal genome to worldwide distribution and realizing that they were more similar to some people than to others. So non-Africans have 1 to 4% of Neanderthal genome in them. That's a lot. We have 30,000 genes. It's a large portion of genes. But every population doesn't have the same Neanderthal genes. So some populations have some aspect of the genome, other ones have other aspects of a genome. So actually 20% of the Neanderthal genome is represented in modern people living today. Again, that's quite a lot, given that the first hybridization event is going to be, what, 50%, and we still have that amount that's retained. Some Asian and oceanic populations have Denisovan DNA. So this is where Denisovan plays into the population. They seem to have hybridized specifically with certain populations of people that ended up becoming South Asian and oceanic people today as they were migrating through that part of the world. We also have some evidence, which I didn't talk about, of ancient gene flow between lineages within Africa. So it's not based on ancient DNA evidence, but on all sorts of models for interpreting modern DNA. So even within Africa, there may have been lineage divergence and gene flow as well. I like this picture quite a lot because it shows, just for the Neanderthals and the Denisovans, where this admixture, how this admixture is distributed. So this, let's see if you can see that. This goes basically from 0 down in the gray, or less than 0 down in the gray, up to about 1.6% in the red. And you can see the various populations around the world where Neanderthal influence is. And it's a little bit counterintuitive that you have less Neanderthal influence in this part of the world where there were most of the Neanderthals, right? But if you think about that, you still have African populations coming out of Africa. So that's continuing to get diluted down, if you will. And the influence was much greater in other parts of the world. This is the Denisovans. So you can see that it's the Southeast Asian and Oceanic populations over here that have, as they were migrating over, picked up those aspects of the genome and moved into those parts of the world. So what specifically has this done to us? Well, the first thing people started looking at was, first they want to know what it says about Neanderthals, really. So we have all these Neanderthal genomes, and what does it say about Neanderthals? Well, it says a lot of things about Neanderthals. We know that Neanderthals now, or at least 2 of them that were analyzed, have red hair. So we're getting some of that pigmentation was in Neanderthals. We know that they were lactose intolerant. That's not really surprising, since the primitive condition, if you will, for people and for all mammals is to be lactose intolerant. So it hadn't evolved yet. We know that they had a gene variant related to brain growth that is different from modern humans. Again, not all that surprising, because Neanderthals actually have bigger brains than modern humans do. And we know that they had, among many other things, I've just plucked a few out, differences in a gene that's responsible for the development of the skeleton. Again, this is not, that one is not a huge surprise, because Neanderthals do look different skeletally in many respects. So that was kind of fun to learn about Neanderthals. But what does it mean for modern people that are living today? Well, like I said, the earliest piece of evidence we have is for movement, and I struggle with the word modern humans anymore in this context, because Neanderthals are part of us too. So I'm using the word African here. As the Africans were moving out of Africa and they were in Asia, and they interbred with Neanderthals at around 100,000 years, or that's where our evidence comes from it, we find lots of things. We find lots of genes that we don't know what they do. We find, of course, there are going to be lots of genes that are neutral, but some genes that we do know what they do, and some genes that seem to have positive effects and some that have negative effects. So, for example, Africans gave to Neanderthals genes that are known to be involved in liver development. So for some reason, those have continued to be here today in modern groups, or that were given to Neanderthals, if you will. Africans gave to Neanderthals, that we can see in the Neanderthal genome, sorry, I misspoke there, a segment of a gene that is responsible for me doing what I'm doing right now, so speaking very quickly. So a gene that's been associated with the ability to have complex communication skills. We know a lot more about what Neanderthals gave to these African populations who ultimately became us, because we have a lot more individuals from whom we've gotten this information. And they really, the really important ones, if you will, from an adaptive point of view, fall into 2 groups. One is skin and hair phenotype. So as Africans were leaving Africa and acquiring genes from Neanderthal, they were retaining ones until modern times that were associated with things like light skin pigmentation, freckling, the cells that produce skin cells. 70% of Europeans actually have a variant that was acquired from Neanderthals in terms of skin pigmentation and freckling. There are lots of skin-related genes that have been acquired by these Africans or modern humans that we don't fully know what they're for, but we can imagine that they may have been related to all sorts of things that skin does, right? It's good for helping you to produce vitamin D, and that would be essential in a northerly climate where it's beneficial to have lighter skin. Pathogen defense, wound healing. It was a rough life. It was a very rough life for Neanderthals in that part of the world. Not becoming too dehydrated. UV radiation plays a big role in here. So again, a dark pigmented skin was not an adaptively great thing to have in a European context, and so it was acquired to have a lighter pigmented skin. The other class that we see of genes that we see that have been acquired by these Africans moving out from Neanderthals are genes related to immunity. So genes that provide a benefit in a context where the pathogens that are there, where the bacteria that are there, where the parasites that are there are quite different from the environment that you were in before. If you look down here, this one particular gene that affects the immune system exists in a Neanderthal gene, exists in 5% of living Eurasians, but exists in 54% of Papuans. So there are certain populations that have acquired these and have retained them at extremely high levels. From the information of Denisovans into these migrating out Africans. What are we getting from there? Well, the two major ones that we know about are, again, genes related to immunity. So pathogens, bacteria, fungi, again, trying to go into a new and different environment. And this one. And this is a really special gene that enables modern-day Tibetans to live at high altitude, low oxygen environments. And those modern-day people acquired that from these ancient Denisovans. Now, one of the really interesting things, as I said, there are neutral genes. There are lots of genes we don't know anything about. There are some we know a little bit about. There are some that we know that are beneficial. There are also some that actually, in modern-day contexts, aren't good at all. So certain introgression of genes into modern people appear to have been selected against. There are all sorts of algorithms that people have done to try and model this. There appears to be some evidence that Neanderthals, possibly Neanderthal males, are a little bit less fit. And probably most sort of excitingly, if you will, there are a lot of genes that are in modern people today from Neanderthals that are implicated in really bad diseases. So things like autoimmune disorder, prostate cancer, cirrhosis. Remember, the liver comes back into the question again, right? Type 2 diabetes, depression. But if you think about it, these are diseases of modern people, right? These are diseases of our modern day lifestyle. For people who live longer, who drink, who eat poorly, and all of these sorts of things. So we don't really know whether these genes, when they were acquired and retained in modern people, were beneficial in the past, and now are not, given what our modern lifestyle is. So what's the take-home message? I love these pictures. This is one of my favorite. I love these ones in particular. These are, it's perfect because we've been doing the Olympics. These are elite athletes of, you can imagine, all different shapes and sizes, because you can be elite in different things depending on what your body type is. And then obviously variation in modern people's faces. So the take-home message here, essentially, is that our history, not just over the last 100 years, not just over the last 1,000 years, not just over the last 10,000 years, but potentially for millions of years, has been one of migration, interaction, and gene exchange. And that as a result of this, we are all, in different ways, hybrids. And it's very difficult, impossible, it always has been, to parcel us into discrete units. It's just not the way that we've evolved. We've never been separate groups of people. We've always been admixing the whole way through. And it's a real lesson, I think, for thinking about modern people today, that our adaptive success, our ability to move around the planet and to be such a widespread, diverse, and successful species is because we have always mixed with each other. So that's my take home, and I'm happy to take any questions. Ooh, that's grim. Okay. Silence. Yes?
That is fabulous. I just want to go back to the dominant model. So about 2 million years ago, that first migration must have been a Homo erectus migration.
Yeah.
So the descendants of that, the Neanderthals, the Denisovans in different parts of the world. Then comes along the modern migration, 70 to 100,000 years ago. And there was no replacement, but there was probably integration. And then what? Kill them off? You had Homo sapiens.
So one thing I didn't really point to, but I should have, is this. So this picture was sort of the sister image to the first one I showed. And what this is showing here is population sizes. So these are effective population sizes. They're not actual population size. The actual population is quite a bit bigger, but you can kind of think of it as the group in the population who is actively breeding at that time. It's a fairly good estimate. And the reason I brought this in is because what it's telling you is that the African populations are and have always been bigger. And that these other populations, because these are hunter-gatherer groups, right? They're really, really low-density groups around the landscape. These other populations are actually quite small. So when you had that second wave, or frankly, it's probably multiple waves of people continuing to move out of Africa. Most of the evolution was in Africa. Africa was probably the densest, and people were continuing to migrate out. They weren't, I wouldn't say they're killing them off. They're just, the gene exchange resulted in everyone just absorbing everyone else, essentially, because of these small population sizes.
So if you say the last Neanderthal was alive 28,000 years?
Well, there are Neanderthals alive today, depending on how you define it, right? If you had your genome done. So.
But it's amazing there wasn't a backflow, so the African doesn't have that 4%. It's all in Europe.
I think it's a population pressure, but I wouldn't discount it because we don't have ancient genomes from Africa. Okay, I'll give you an inside thing. We do have ancient genomes from Africa, but they're not published yet. So, yeah, I mean, outside of that 4,500 one that's from East Africa, but we don't have deep ancient genomes from Africa. So, our window into this is actually quite narrow, I think. Yeah.
So I don't want to hog this. This is the last one. Sebastian Harbour. Apparently, he's got him down to fine art. You give him anything, and he can actually.
There are a handful of labs in the world who are able to do that. His lab, Eskaviller's lab, there's the lab in Sweden. There are really just a few. The amount of technological development that you have to do this ancient genome work is remarkable. I wouldn't, frankly, recommend anyone start up a lab because it's so much money, but also intellectual, capacity in those spaces. But yeah, they really do. Yeah.
Thank you.
Yeah.
You talked about the papillons being affected by the Denisovans and the Neanderthals. Why would they be the center of the universe for everybody to come into?
Probably has to do with migration. So when you have these, they were basically passing through the area and taking the genes with them. And because they're so isolated, because they're so at the edge of the world, if you will, they haven't had that later day influence to alter it in the same way. Yeah.
It's striking that geographic distribution of Neanderthal, how far they got.
Yeah.
Down to New Zealand.
Well, it's Neanderthal genes, right? No, you're talking about the wrong way. This. Yeah, so this is Neanderthal genes that are in. So the reason why this is kind of reverse, so this comes back to the other question, is that when you have this population pressure and people are migrating out, eventually migrating around and coming into the new world, right, if you wrap that around a globe, you're going to have this constant, where they're isolated, they're going to save or keep, if you will, more of the genomes that they kind of picked up on the way, and then not have new influences from the other populations because they're more isolated. So it's kind of, that's why I was saying it's kind of the opposite of what you would expect, where you have these isolates at the periphery that actually have the higher level of Neanderthal ancestry, and the ones that are at the core that over time have gotten diluted, because these are modern populations we're looking at on this map. And a lot of things have happened since. The Neanderthals. Well.
I mean, does that not favor a dominant, fitter race just extinction?
No, because of course, you have light skin. Your ancestors evolved in Europe. If you hadn't had Neanderthal contributions to your genome, which probably most of you in the room do, you wouldn't have been as evolutionarily successful. They did not go extinct. Their genomes were absorbed and promoted the evolution of our species and our diversity. Well, we all. We also don't have anything that looks like an ancient African around. You know, if you went to some of the East African material from 150,000 years ago and that person was walking down the street, that would be pretty remarkable too. So, you know, we as a species have changed in the last 40,000 years and become quite gracile, and we all kind of look this way. And both of those things would be really surprising, I think.
I find that slide of yours with the tree on one side and the sort of amoeba-like conglomeration on the other side the most enlightening slide, because we've all grown up with that tree in mind, and that other one changes the whole idea.
Mm. It does, doesn't it? Yeah.
On that, so how is this going to go forward? In 50,000 years' time, what's going to be the big tech?
I always get the question about future human evolution. I mean, the reality is a lot of this stuff breaks down in modern times, right? Because we can move all around the world, and we can use our culture to buffer us from the environment. So you don't have, okay, you do have, if you have very dark-skinned people moving to very northerly climates, they can have real problems and have to take vitamin D supplements because they can't produce enough vitamin D because their skin is low. But we can do that, right? All the light-skinned people living in high UV environments can wear sunscreen. All of these sorts of things. So I think in going forward, our culture has provided such a buffer that these effects just won't be as clear. It's not that selection doesn't continue to act on us. It's not that we don't continue to evolve, because we do. It's just that you're not going to have the same kind of response, if you will, to the environment in people that live today that we can map here in the past.
Maybe it'll be Martians doing a reverse migration back to...
Well, I can say it's very likely that we'll all end up medium brown in the end. I mean, given migration and gene flow over time, that's one thing I can probably guess. Unless the ozone layer changes, and then we're all in trouble. Yeah?
Can you say something about genetic drift?
Can I say something? I can say a lot about genetic drift, actually. It's my other favorite topic. Obviously, drift is a big player here as well, right? Because, okay, so genetic drift is essentially, you know, it's the reason why we can apply a molecular clock to things and date things. It's because a vast majority of the genome just sort of changes through time and diversifies. And when you have small populations in particular that can play a very large role in diversification, because little populations will end up just losing variation by chance, or variation will get fixed in populations, and it could differ from population to population through time. So they're kind of random chance effects. And genetic drift has played a very large role across the evolution of Homo. I didn't talk about it today, but a lot of the variation that we're seeing is also because of drift effects, as we have these small groups moving across the landscape. So chance. But that's another, it's a separate, yeah. It's not separate, it's actually all related, but it's another topic. Yeah.
You said there was no Neanderthal DNA found in mitochondrial DNA, or there was no...
Yeah, no Neanderthal mitochondrial DNA found in people today.
In people today. And has there subsequently, has it been found? Or has there been a blockage somehow in the female line that wasn't passed on? Or is there any thought as to why that was?
So there's some ideas about Neanderthals, you know, the females being the ones that were able to breed, and that essentially the human lineage, if you will, again, swamped them, that maybe the males were less fit. But I don't know if there's a good theory out there for why we wouldn't have any, unless, and I'm just making this up off the top of my head, unless the mitochondrial DNA of Neanderthals were in some way less compatible to the environment, the cellular environment that was the result of this hybridization. Yeah. One last one? I can take two, doesn't matter. Go ahead.
Getting back to the time when there's migration from Africa into what we now know as Europe, this African group encountered a unique group called the Neanderthals. Where did they actually originate from? Why were they there?
Everything that was everywhere else in the world originally originated from Africa. That's the other kind of take-home message of all this, right? We're all ultimately Africa. It's just how far back in time we go. Because our lineage as a whole, all hominins originated in Africa. So they evolved from migrations out of Africa and adapted. It's just that they had adapted for a much longer period of time in Europe. And so in some ways you can think about the gene exchange as a shortcut to adaptation, so that when people came out again later, it was a lot easier, quote unquote, to mate with the Neanderthals and pick up their genes and have their skin color change really quickly than it was to, have natural selection act on them over a long period of time in that environment. So that's one of the powerful things about gene exchange, is that it can facilitate very rapid evolution through this movement of genes.
Wasn't that related to the Ice Age as well, in the sense that the Ice Age covered quite a large area, which meant it implies that the population was pushed out. But as the Ice Age retreated or receded, then there was a flow back again.
Well, there has been quite a lot of movement back and forth in that part, as there have been these cycles, these sort of glacial cycles. And that's part of why I was saying that the Neanderthals generally lived in a really difficult environment. And it is possible that the Western Neanderthals, and we don't have great information from Western Neanderthals, we don't have genomes from that group. And we do have some mitochondrial DNA. It is possible that the Western Neanderthals were more isolated. And it is possible, again, that pockets of them did not contribute to modern diversity as we see it today. So it's a really incomplete picture that we have right now in terms of the extent, if you will, of this kind of phenomenon. There was one more question over here, yeah.
Is there any evidence of continuing evolution in terms of increasing brain size?
No, if anything, we're getting smaller. Yeah.
Otherwise, in 100,000 years, you could only be born by caesarean section. I guess there was also a change in the pelvis.
Yeah, so the caesarean section thing is a different question, right? So culturally, we're allowing for more variation in lots of things: brain size, birth weight. Birth weight is actually a bigger issue right now than brain size, because of our modern diet. We have very fat babies often. And caesarean allows for us to have those very fat babies, which previously would have probably killed the mom. And also very premature babies, which previously would have killed the baby. So yes, in a very real sense, again, this is a nice example of how culture can allow us to, allow things to go through, if you will, that maybe wouldn't have in the past. I shouldn't say go through when I'm talking about babies.
Just a quite different question. Is there anyone who particularly studies all the Persian peoples? Because they've got masses of different gene variations, apparently, in groups living close together. Now that modern civilization has arrived, they're presumably mixing much more. What's happening to their genes? Is there anyone who looks at
I'm sure there are. I'm not particularly familiar with the diversity within their genes, but there are large projects of people looking at genomic diversity across the planet, just like there are here in Africa, where you have these massive modern human diversity projects.
But there seems to be so much diversity in one
Yeah.
tiny place there.
Yeah.
It seems to be worthy of a special study.
Yeah.
Could there have been, you pointed out that there was integration between different, what we would consider species, but actually
Lineages, that's what I use.
Right. Now, could that have resulted in different, as people moved up and they integrated with different lineages in Europe or in Asia?
Could that have
resulted in a different Denisovans?
Yeah, we don't understand the origin of Denisovans at all, really. The only thing we have is just these few individuals, and we don't know what they look like. We don't know anything about it. And in fact, the fossil record across that whole part of the country is problematic. It's sparse. Certainly when you get into the Far East, it's often very poorly dated. And so it's kind of understanding the emergence of those groups is pretty limited. Yeah, particularly Denisovans.
According to that sort of, what I call amoeba-like slide of yours.
That's my picture of what it, yeah.
It actually indicates a very interesting possibility of cross contact and integration.
It does, and
all sorts of
it's the model to be tested essentially right now. Yeah. Okay. Thank you.